Thank you for that introduction. I'm excited to present to you some of our preliminary work looking at the effects of maternal smoking in adults using the UK Biobank. So to provide some background, the prevalence of prenatal smoking worldwide remains high in several countries, particularly in Canada, Australia, and several parts of Europe. And although prevalence estimates of early postnatal smoking has been relatively understudied, there is evidence that these rates are at least as high as those that occur during the prenatal period. And this is a major public health concern because there are over 7,000 chemicals that have been identified in cigarette smoke. Over 250 of these have been identified as toxic, and more than 70 have been identified as known cancer-causing agents. And the image here comes from the CDC, which shows just a handful of the harmful chemicals that are released in cigarette smoke. The vast majority of toxins in cigarette smoke are actually released into the atmosphere, and this is what contributes to the harmful effects of secondhand smoke. Children and adolescents are far more vulnerable to the effects of secondhand smoke than adults, and this is partly due to the increased respiratory rate in children and infants, which allows them to consume greater amounts of smoke and greater amounts of toxins. Additionally, their underdeveloped lungs and underdeveloped immune system puts them at an increased risk for disease later on. And this is consistent with the developmental origins hypothesis, which is a body of work that basically is dedicated to showing how early exposures to environmental stressors actually prime for later life disease. And we see that this is true for individuals that are exposed to maternal smoking. There are higher rates of childhood and adolescent illness. In children and adolescents, we see that there are increased rates of ear infections, respiratory conditions, and visual problems, as well as neurodevelopmental conditions such as ADHD and autism. In adults, there's a significantly higher risk for cardiovascular disease and psychiatric illness among those who are exposed to maternal smoking around birth. And so to apply this model to prenatal smoking, the idea is that exposure to maternal smoking around birth sets the stage for this cascade of problems that can both directly and additively affect brain structure. Previous neuroimaging studies have shown that there are structural brain abnormalities in children and adolescents who've been exposed to maternal smoking. These studies collectively show that there are widespread abnormalities in the cortex, primarily in cortical thickness, and then additional work has shown smaller volumes in the pallidum. There are other studies that have shown additional effects, but these are the effects that are specific to early life and perinatal cigarette smoke exposure. So while we know that there are these abnormalities that occur in children and adolescents who have been exposed to perinatal smoking, no study has examined the potentially long-term effects that could persist into adulthood. So the purpose of our study was to identify structural brain signatures of maternal smoke exposure in a large population-based sample of middle-aged and older adults in the UK Biobank. And we hypothesized that exposure to maternal smoking would be associated with brain abnormalities relative to unexposed adults, specifically that we would see a lower cortical surface area in regions of thalamocortical projection zones, and also volumes in dopamine-rich structures of the basal ganglia. And we specifically formed these hypotheses first because surface area is particularly vulnerable to early developmental exposures and also expands rapidly during the first postnatal year. And there's also a large body of work showing that prenatal nicotine disrupts cholinergic and dopaminergic activity in regions that are densely populated with nicotinic acetylcholine receptors. So we examined 9,165 adults from the UK Biobank, and these data come from the July 2017 data release. Ages range from 44 to 80 years old. The vast majority of the sample was white, and 4% were current smokers and 34% identified as previous smokers. Maternal smoking was assessed via touchscreen, and participants responded to the question, did your mother smoke regularly around the time when you were born? Of the sample of 9,165, 2,663 responded yes, that they had been exposed to maternal smoking. All imaging acquisition was completed on a single 3T Siemens Skyra. Cortical thickness and surface area were estimated in parcellations of the Desikin-Kilyani atlas using FreeSurfer version 5.3. And all of the quality control procedures were completed using protocols from the Enigma Consortium for all 68 bilateral structures. And this QC process involves visually inspecting all regions for over and underestimations, and segmentation issues that were confined to localized regions were included in the analysis after we removed the affected ROIs. And then subcortical volumes were extracted directly from the UK Biobank database, 14 bilateral regions, and these were processed from UK Biobank researchers using FSL first. Preliminary analyses were conducted to determine the need for covariates beyond age, sex, and intracranial volume. And so we specifically explored differences between exposed and unexposed groups in race, and this was dichotomized to white versus other races, given that more than 99% of the sample identified as white. Education was also dichotomized to a college degree or less, and this was our primary measure of socioeconomic status. And then smoking status in adulthood, we examined current versus previous and never smoking, and whether that differed between exposed and unexposed groups. And chi-square analyses showed that each of these variables did differ between exposed and unexposed groups, with the exception of previous and never smokers. Those did not differ, so we used current smoking as our primary covariate for the main analyses. For the analyses, we used multiple regressions that were separately run for cortical and subcortical measures. And each of these regressions we covaried for intracranial volume, race, sex, age, education, and current smoking status. And the false discovery rate procedure was applied to all models. Our results revealed that there was significantly lower cortical surface area in several regions throughout the cortex, with a predilection for effects in mid-posterior brain regions. We also observed significantly lower cortical thickness in the temporal lobe, and significantly lower volumes in regions of the basal ganglia and the amygdala. Across all these analyses, the effect sizes were modest. The strongest effect sizes were actually observed in the bilateral pericalcrin, inferior parietal cortex, middle temporal gyrus, and the left cuneus. And these regions are particularly noteworthy because they're all involved in both primary and secondary visual processing streams. As a post-hoc analysis, we wanted to see if we would observe these findings or similar findings in a subset of neurologically healthy individuals in the UK Biobank. And to do this, we excluded individuals that had documented or self-report medical or psychiatric conditions with known effect on brain structure and function. And this left us with 5,077 individuals in the UK Biobank. And after we reran the same analyses, we again found that there was significantly lower cortical surface area in these primary and secondary visual processing regions. And interestingly, the effect sizes in the neurologically healthy adults were slightly larger than those observed in the whole sample. However, we did not observe any group differences in cortical thickness or subcortical volumes. So to summarize these main findings, maternal smoke exposure was associated with abnormalities in cortical surface area, with the strongest effects in regions that subserve primary and secondary visual system functions in both the whole sample and then neurologically healthy adults. This is consistent with several studies that show that there are increased visual disturbances in children who were perinatally exposed to smoking. And it's also consistent with several animal and experimental studies showing that prenatal nicotine is associated with increased acetylcholine receptor binding in regions of the visual cortex. So this is consistent with our cholinergic hypothesis. And there's also additional work showing that exposure to mercury, which is one of the neurotoxins in cigarette smoke, is linked to cholinergic changes in the occipital cortex and basal ganglia. And obviously, we can't speak to whether or not the findings in the study were specific to any certain chemical in cigarette smoke, but this is certainly an intriguing area of research, of future research. So limitations of this study, there are several limitations related to our measure of maternal smoking. It's very nonspecific, so we don't have information on the timing of exposure, whether it occurred pre or postnatally, or how long exposure occurred. We also don't have information on paternal or familial smoking during early life. This is obviously an important period of brain development. And we don't have exposure to polysubstance use during early life. And then finally, socioeconomic status, which is highly related to maternal smoking, is unknown early life in these adults. We do have analyses underway to examine whether birth weight is a mediator between smoke exposure and brain structure. We're particularly interested in looking at this because low birth weight has been identified as an established outcome of maternal smoking. And then we're also looking at maternal smoke exposure as a potential moderator between brain structure and cognitive function. I'd like to thank the UK Biobank for allowing us access to this amazing database, also the NIH for supporting our Enigma pipelines, and then all of the co-authors that have put in a lot of work towards this project.
Thanks, Lauren. And